Hi, my name is Madison Poole. I'll be answering the case 1 as well as the case 2, and I'll be reading from my script on the side of the screen, so that's why my eyes will be moving back and forth. So case 1, there's 2 questions. Under normal conditions, describe all the ways in which the pyruvate kinase is regulated within the cell. So the enzymatic activity of pyruvate kinase is regulated by both metabolites and intercellular signaling pathways. Pyruvate kinase integrates metabolic inputs and signaling to modulate glucose according to the requirement of the specific cell. The pyruvate kinase activity is reduced by phosphotyrosine growing signaling. The pyruvate kinase activity is reduced only when growth signaling is active within the cell, and this effectively coordinates glucose metabolism with tyrosine-mediated signaling. The activity of this enzyme can also be reduced by the oxidation of enzymes C358 by ROS, or a reactive oxygen species. Under nutrient-depleted conditions, the acetylation of enzyme K304 leads to lowering affinity of pyruvate kinase for PED and degradation of this enzyme by itself. The second question to case 2 states that this logic seems counterintuitive for a glycolytic tissue, and what is the rationale for this mechanism, and what other pathways may cancer cells be diverting substrate to? So this rationale for this mechanism is due to a specific enzyme. Now, this key enzyme in cancer metabolism is called PKM2, or pyruvate kinase type M2. Most cancer cells have increased expression of this certain enzyme. In rapidly proliferating cells like embryonic and adult stem cells, a family of regulator genes known as MYC increases the expression of glucose transporter 1 and the pyruvate kinase type M2, or the PKM2 as stated earlier. This promotes the Warburg effect, and this effect refers to the fact that cancer cells are counterintuitive because they prefer fermentation as a source of energy rather than a more efficient mitochondrial pathway of oxidative phosphorylation. MYC is oncogenic transcription factors which are overexpressed within cancer cells. The MYC is associated with the promotion of RNA. MYC is thought to induce the expression of splicing factors like PTB, which favors splicing of the PKM2 enzyme over the PKM1. MYC stimulates glucose uptake and glycolysis. MYC also induces glutamine consumption and metabolism, which appears to be important in glucose of oxygen-deprived conditions. Now I will be moving on to case 2, and there was no other answer choices that I needed to defend on case 1, and it's the same for case 2. Now, the question concerning case 2 is, how does glucose uptake in normal tissue compare to glucose uptake by tumor cells in the above image with an MRI and a PET scan? And why would this be advantageous to the invasive tumor? And the following question, what modifications may have occurred in cancer cells to alter glucose uptake as predicted above? So, glucose as a hydrophilic molecule needs carrier proteins to cross the membrane. 2 families of membrane proteins participate in glucose uptake. One is the family of facilitated glucose transporter proteins. These proteins move down a concentration gradient, and they are known as GLUT proteins. The other family is sodium-coupled glucose co-transporter proteins. This process requires energy, and it is done by SGLT proteins. The GLUT proteins are constituted by a family of proteins with similar functions but with a differential distribution according to the type of cell. In normal tissue glucose, it is regulated in liver by family members GLUT2, GLUT8, and GLUT9 proteins. And the skeletal muscle, it is regulated by GLUT4, GLUT11, and GLUT12 proteins. In the brain, it is regulated by GLUT3, GLUT6, and GLUT8 proteins. And in the adipose tissue, it is regulated by GLUT4, GLUT8, and GLUT12 proteins. Now, it is known that glucose uptake across cell membrane is a rate-limiting step in glucose metabolism in normal cells. But for cancer cells, they need to adapt their metabolism to survive and multiply under tumor environment conditions versus the normal conditions. As they have lost control over the proliferation process of the cells, they need a constant supply of nutrients and a constant supply of energy. And the cancer cells achieve this by glycolytic process, which is around 30-fold higher than normal cells. So they must increase glucose uptake rate very significantly. In fact, in cancer cells, it has been demonstrated that an increase in expression of the GLUT proteins showing abnormal patterns compares with healthy cells, so this event allows them to optimize their energy supply, which is advantageous for growth. In normal cells, the glycolysis pathway is part of mitochondrial respiration process, which results in the gain of energy as ATP molecules from the breakdown of carbohydrates in an aerobic way. But on the other hand, cancer cells switch the glycolysis process, even though it is a more inefficient way of gaining energy. Moreover, in this cell, glucose decreases oxygen uptake, resulting in a respiratory impairment using a primitive, inefficient reaction to generate the high quantities of energy that's needed. It uses aerobic glycolysis, which results in 2 ATP, glucose instead of 36 ATP, and glucose as a result of mitochondrial respiration. And this explains why this cell's overexpressed glucose membrane transporters increases the uptake of glucose because it needs to compensate for this deficiency. And there are no answers to defend on the case 2, and that is it for my answer. Thank you for watching.